The eLife podcast from eLife, the open access journal for outstanding research in the life and biomedical sciences. Online at elife.elifesciences.org.
Hello, welcome to episode 5 of the eLife podcast, which is produced by The Naked Scientists. I'm Chris Smith. And this month, before we hit the research papers, congratulations are in order. Apart from celebrating eLife's first birthday on October the 15th, there were also not one, but 2 Nobel Prizes. One for eLife editor-in-chief Randy Sheckman, who shared the Physiology and Medicine Prize for his work on vesicles, and the other in, as the BBC dubbed it, complex chemistry, to Michael Levitt, who's on the eLife board of reviewing editors. Now, here are some scientists who might be future Nobelists in waiting. In our lineup this month, the discovery of the potassium channel that controls the activity of the human sperm, the cyanobacteria in your intestine that turned out to actually be something else, and the workings of the stem cells that populate the human airway. What does smoking do to them? Now, we know from work in mice that sperm use certain ions, like potassium, to control their activity. And while ion channels to admit potassium have been found in mice, no one had found the human equivalent. Until now. Nadia Manovitz.
We wanted to identify a potassium ion channel in human spermatozoa. Sperm are highly differentiated cells, and their task is to carry genetic material of the father, fuse with the egg, and produce offspring. And to do that, they depend on several chemical compounds, such as electrolytes, sodium, potassium, calcium, magnesia. These compounds cannot enter or leave the cell just as they want. Sperm cells have to have pores in their membrane, and these pores we call ion channels. 2 of such ion channels have been identified, a calcium channel, the other is a proton channel. And both of these channels are dependent on membrane potential. And the membrane potential is being set by potassium ion channels. For mice, the identity of a potassium channel has been characterized, but such data were not available for human sperm.
So we know that there has to be a potassium channel on the human sperm because we know it's there in mice, and it's fundamental to how the mouse sperm does its job. So the question is, well, what is it and how does it work? Can we identify it?
Exactly. We addressed this question to apply the so-called patch clamp technique to the cells. This is an electrophysiological technique. You record electrical currents of the cell, which gives you an answer: is there an active potassium channel present in these cells? By applying specific inhibitors, you can then fingerprint the pharmacological profile.
So you took some human sperm and you applied this patch clamp technique, which enabled you to study how ions were going back and forth in and out of the sperm. And you've been able to identify what must be a potassium channel on the human sperm. Is it the same as the one that we find in mice?
No, it's a total different channel. The potassium channel in mouse sperm is called the slow 3 channel. And this channel is activated when the intracellular pH rises. In human sperm, changes of intracellular pH do not activate the potassium channel. In human sperm, changes of intracellular calcium activate the channel.
So this must be some kind of evolutionary solution to the way that human and mouse mating habits differ.
Yes. It could also be, if we look on the genes for that slow 3 channels in mice and the so-called slow 1 channel in humans, they share a certain amount of homology It's about 50 or 60%. And you could think that at some point during evolution, mice and humans diverged from each other.
So, what are the implications of finding this new potassium channel? Does this open up a new ability for us to manipulate the behaviour of sperm or the activity of sperm? And does it also explain why some people can't get pregnant?
Yes, identifying an ion channel helps to understand how male fertility works. So, having identified slow 1 as the principal potassium channel in human sperm, it might open up research on a genetic level to screen men whether they have a genetic defect in the slow 1 coding gene, and then think about perhaps about the proper treatment.
And looking at the flip side of the coin, what about contraception?
Yeah, this is a very good point. When you try to solve questions what could cause male infertility, you could also think about developing a male contraceptive. The findings we have published could be the basis to develop a male contraceptive, but I have to say, if you want to develop a male contraceptive, you have to make sure that this drug only affects sperm or developing sperm cells and not other cells of the body. Slow 1 has been shown to be expressed and conduct potassium ions in nerve cells as well as in muscle cells.
So that means then that it would be quite difficult to come up with a discrete blocker unless you used or exploited some other function of the sperm to target that.
Exactly.
Nadia Manovitz. She's based at the University Hospital in Essen. Forensic scientists have a range of techniques to work out how long a person has been dead. These include looking at things like insects that may have moved in to help decomposition along. Unfortunately, these measures are fraught with difficulties and they tend to be quite inaccurate. But now, University of Boulder, Colorado scientist Jess Metcalf has been looking at another, potentially much more accurate way of doing it. She's looking at the communities of microorganisms that grow in and on the body.
So we know microbes play an important role in decomposition. That's no secret. Forensic scientists have known this for a long time. However, we haven't had the tools to really study the microbes. And because decomposition is driven by biological processes, forensic scientists try to use biological information to estimate the time since death. For example, if you watch CSI or some similar program, you might always see a forensic scientist sampling blowfly larvae. Oh, it's at this stage. This means that this body could have only been dead for so long. And so our question was, can we use microbes to try to answer the same type of question? Because every approach in forensic science for estimating the time since death, they all have some amount of uncertainty around them. And if you combine these different approaches, you can narrow that window of uncertainty. And our question is, can we use the information about microbial community change during decomposition to help narrow this window?
Why should that be better than what the other measures can already offer, Jess?
Every type of approach has its limitations. So, for example, body temperature and lividity, that's only going to be relevant in the first 48 hours. And then things like the succession of insects might be limited by the season. So sometimes in the winter, insects never show up at a body, so you can't use that. However, we all have microbes in us and on us when we're alive, and so we already have the organisms at the time of death.
It's like our own inbuilt biomarker. So how did you do this?
We used a mouse model system. And the reason we did that is because really what this question is if you have lots of replicates, are you going to see the same microbial community change in each replicate? Or is it just some stochastic response of the microbial community? What we want it to be able to use it as sort of a biomarker to estimate the time since death, is we want it to be consistent across replicates. And that's really hard to do in using, for example, donated human corpses, or even what's often used in forensic science is a swine model. those experiments are usually limited to like 1 to 3 individuals. And so with a model system, like a mouse model system, we were able to use 40 individuals, and we sampled 5 at a time over 8 time points. And this was the first time that someone was actually able to characterize that bacterial community change.
Could you also look not just in the body, but in the environment? Because the body, by virtue of breaking down, is going to release various chemicals and nutrients into the surrounding environment, which will in turn surely alter the microbial communities there too.
That's a great question. And that's one of the things that we looked at in this study, how decomposition of a corpse affects the surrounding soil. Oftentimes, in a crime scene, you'll find a body on the soil outside. And so if someone moves a body, can we test the soil and say that something was decomposing on it? And so for the soil, we're kind of interested in 2 different questions. One, can we also use the change in soil microbes to estimate the time since death? But also, is there a really significant signature left by the decomposition process? And the answer to both of those questions was yes. Then at every site, we saw really significant change in both the bacteria and sort of the nematodes and other microbial eukaryotes that were present. Everything changed very significantly.
How were you measuring the microbes? Was this using a DNA technique?
Exactly. So what we did is we borrowed the techniques that are now more commonly used to really understand the human gut microbiome. Take a sample and at the depths of like millions of sequences, then characterize the community of bacterial species and the community of, say, fungi and nematodes that are present.
But obviously, where the majority of the bugs are in a human is inside us, in our guts. And so that's going to really strongly dictate what we then get rotted down by, at least initially, isn't it? So does it make a difference to your assay, do you think, or will it, whether you've got a person from one culture or another culture, one sex or a different sex? Because we all know that our microbial spectrum is as unique as a fingerprint. We're not all genetically identical study mice. So will it still work in a highly genetically diverse and microbiologically diverse human population?
That's a really good question, and that's what our follow-up experiments are getting at. Now we're going to look across different host species. So obviously mice have different gut microbes than pigs or swine than do humans. And so one of the things that we're going to do with all of our different studies is then compare across different host organisms or species that, does that influence what your decomposer community looks like? And that sort of gets at the same question of like, different diets and different sort of gut microbiomes.
Jess Metcalf. And it's possible that some of the bugs she was seeing were from a newly described bacterial family known now as the Melanibacteria, which had previously been written off as cyanobacteria until Sarah Dirienzi came along.
It's been known for a while that within the human gut, there are sequences that appear to be very closely related to cyanobacteria, so closely related that they're generally just assumed to actually belong to cyanobacteria. So whenever these sequences pop up, they just automatically are assigned as cyanobacteria when they're not. And where this really gets confusing is that cyanobacteria are photosynthetic. So they like to be in places where there's light. That's how they live. That's how they get their energy.
And where you're looking, naturally, the sun doesn't shine terribly often.
Exactly. There's no light in the human gut.
Well, not on a good day, anyway.
Yeah, exactly. So when most people read, cyanobacteria in the human gut, they say to themselves, that must be the plants we've been eating. It must be from the photosynthetic cells that are contained within plants, and not that this is some cyanobacteria that's actually growing in the human gut. So what we were setting out to do is to try to actually determine, well, what exactly are these bacteria? And our lab has tried for some time to actually grow them in the lab, and this has proved to be very difficult and unsuccessful. What we did was complete sequencing of fecal samples. I worked especially in collaboration with Itai Sharon, who's a postdoc in Jill Banfield's lab. And they have this really amazing technique which allows them to sift through a pool of DNA sequences and figure out who belongs to whom. So what the method expects is that DNA that comes from one organism should all be in about the same amount, whereas DNA that comes from another organism should be in a completely different amount.
I get it. And so you can begin to assemble the bits of DNA that go together based on the frequency of those components. What did that tell you?
So what that allowed us to do is to get all the DNA that belonged to this weird cyanobacteria-like organism all together. And then from there, we could go and use traditional characterization of genomes based on information we've learned by studying, thousands of other bacteria over the years.
And then it became patently clear this was not cyanobacterium, this was something else.
Yes. First, we firmly established that this is absolutely not a cyanobacteria.
So what actually is it, and what does it do, and why is it there?
So we've termed this new group of bacteria melanobacteria, and we've discovered that it's entirely non-photosynthetic, so it's very different metabolically from cyanobacteria. And instead, it's entirely anaerobic, and it uses fermentation to get its energy. So that means it's breaking down sugars.
But given its stark differences to cyanobacteria, why is it at the same time so similar to them?
Right. So there's a specific gene, the 16S ribosome. This is the best marker that we have to understand evolution. And so by following that, we see that it is closely related to cyanobacteria. So what we hypothesize is that at one point there was a general ancestor, and that then it either went off into the light, so to speak, and figured out how to use light for energy, or it ended up burying itself in dark places like the human gut. In those environments, it had to just rely on fermentation entirely.
So if we look outside the human gut, where else would we find this organism, this family, and what sorts of things does it do in the environment or in other environments other than in us?
So it's also found in water systems. We specifically were able to sequence one out of an aquifer. What exactly it does, we're not 100% certain of. We have some ideas of what it does in the human gut. We're assuming metabolic similarities to a group of organisms called firmicutes. So firmicutes act as major fermenters in the large intestine, where they break down sugars and can convert them to short-chain fatty acids. And we think these melanobacteria can carry out a similar process.
Is there any evidence that they may be linked to pathology, or are they thought to just be passengers that are harmless?
Yeah, we think they're actually, if anything, beneficial. They're also able to produce the B and K vitamins, which the host does need to get from its gut bacteria. And there's some evidence based on its interaction with the immune system that it would not be a pathogen.
So what questions now remain outstanding? You've found this organism, you've proved what people have missed all along, but now what are you going to do?
Oh, we'd love to know what it looks like. We haven't seen what a cell of it looks like yet.
So apart from trying to grow it and look at it so you can have pretty pictures of it and publish the first EM of whatever this is, what else are the big questions you're going to ask of it genetically and evolutionarily speaking?
So we'd like to get a better idea of why it's enriched in herbivores, for instance. So one of our findings is that it's enriched in mammals that are herbivores.
Does that include human vegetarians?
Unfortunately, there's not enough data for us to be able to really answer that question. However, we were able to compare humans living in more Western cultures versus those in Venezuela and in Africa. And we found that they are more present in the people from South America and from Africa, which are also on more vegetarian diets. But we don't have that 100%. Yes, if you eat your vegetables, you'll end up with melanobacteria in you.
Can't see supermarkets rushing out to use that slogan somehow. That was Cornell's Sarah Dirienzi. You're listening to the eLife podcast with me, Chris Smith, from The Naked Scientists. Still to come, why it's not just humans that have analogies like being top of the pile or bargain basement. Chimpanzees do it too. But before we find out how, take a deep breath and ask yourself, where do all of the cells that make up my airways come from?
So my name's Sam James, and I'm working as a Wellcome Senior Fellow at University College London. For some time, many people have felt that the organs of our body rely on stem cells to repopulate all the cells in our organs when we need them. So these stem cells should be capable of producing daughter cells of lots of different types. Now, most of this has come from data from mice, where it's possible to track individual cells over time. But this has never been possible in humans. So we were looking to see whether stem cells really existed in the airways. And in the lung, there are cells which secrete mucus. There are cells that have little cilia on them. And there are basal cells, which are chunky little cells which sit right at the bottom on what we call the basement membrane, which is like the pavement of the lining of the airways.
And those chunky cells, are they the stem cells?
From mice, it has been felt that basal cells are stem cells of the airways. So I suppose that was what we were expecting to see with our work.
And now tell us how you actually did this.
It's quite a challenge in humans because what we actually need to do this is to be able to see a stem cell and then see how it divides and look at what we would call its daughter cells to see what type of cells they are. And to do that in a human is pretty well impossible on any internal organ in particular. But what we found was that there was a scientist, Sir Nicholas Wright, at Barts in London, and he had worked on a way of staining and seeing individual cells for an enzyme that's produced by the mitochondria within our cells. And what he found actually was if these mitochondria had some sort of genetic mutation, which just actually occurs by chance, if those mutations collected, eventually an individual cell would stop expressing that enzyme. Now that means that with this stain you can see that cell, and you can also see daughter cells which have come from that individual mutation.
Because they also carry the same mutation and therefore will have the same funny staining pattern.
Exactly.
So you can use this unique labelling of cells in order to track individual lineages of cells.
That's right. So when we look down the microscope at these cells, so these are in patients that have undergone some sort of lung surgery and had an area of lung taken out, we put the stain on so we can see these cells that are lacking the enzyme. And when we actually pluck out these cells individually and test their DNA, we can prove that they are indeed all related.
And returning to the question you started with, which is what do these stem cells turn into, what do you find when you examine all of those clones of cells that have come from one source stem cell?
Well, what we found was actually pretty interesting. So the individual clones of cells were all identical, and within them they had lots of different cell types. But there were two really important things. One was that every cluster had a basal cell, whereas other cells were sometimes missing from these clusters. So that means that the basal cells are indeed the progenitor cells. But what was really intriguing was that when we showed the results to a mathematician in Cambridge called Ben Simons, he looked at these and said, actually, these aren't related. And in fact, what these clones are doing is entirely by chance. So in fact, these aren't pre-programmed stem cells that are doing a particular job, but in fact, basal cells that are just dividing by chance. And so they may divide to reproduce themselves, they may divide to become a different type of cell type, or they may just die.
So what are the implications of this?
There are a number of implications. The first is, as many of us know, there's a great deal of research looking at regeneration of organs, and there's been a feeling that you must get the stem cells and understand what the stem cells of an organ are to be able to regenerate it. Our data from the airways of humans suggests that that isn't the case. You simply need to collect a broad population of these basal cells. The second thing that was quite interesting was that we looked at non-smokers and compared them to smokers. And what we found in smokers was that the whole process is basically speeded up by 2. Now, this is quite important because, one, it suggests that the airways of smokers are aging twice as quickly as they should. But the second thing is these clones are expanding twice as quickly. So if you ended up with a clone with a genetic mutation which may contribute to forming a cancer, then that clone is already spreading twice as quickly.
But going back to your method, is it possible that what you're seeing is an artifact of the fact that the cells have a mutation?
Yeah, so that's a really important point, because you're quite right. If the mutation led to some sort of advantage for these cells, then they may be expanding more quickly than we would expect. But quite a lot of work has been done with these mitochondrial mutations, and these mutations aren't thought to affect the way the cells proliferate or survive at all.
UCL's Sam Janes. And finally, why do we use phrases like top tier or underclass when we're considering our peers? Some people think it's because we have language and therefore we use verbal metaphors as a convenient way to map priorities or social positions. But then what would happen if you asked a chimpanzee, which of course can't talk? Ikuma Adachi.
What we try to find is the evolutional origin of the language. And especially when we use a word high, low, those words can be everything, right? The price is high and low, and space, of course, is high, low. So why this happen? We tackle this phenomenon in non-human animals because this phenomenon is often argued in a context of the language evolution. Because we evolved this language system, we have this systematic mapping.
So this would be like a sort of social pecking order. There's another metaphor, isn't it? Where you've got people who are regarded as the top of the tree and the person who's at the bottom of the tree, or premium price versus bargain basement.
Yep.
And you're saying that we have those metaphors only because we have words for them.
That's what a lot of people actually believe. And what we raise the question here is it really so? This might reflect more simple another mechanism to reduce the information processing. So in that case, other species should also have this kind of a connection. So that's what we try to reveal first.
So your argument would be then that actually having that concept of high and low, social pecking order or whatever comes first. There's some kind of neurological correlate for that, and we humans have just adopted some words to put that into words, whereas other animals, simpler animals that may not have language, nonetheless would have that kind of construct still of a concept of things being higher or lower than others.
Right, exactly.
So how on earth can you probe that?
So what I did is quite simple approach, asking if non-human primate chimpanzee also have this kind of mapping or not. So we tested 6 chimpanzees, and what we did is showing one picture first of the certain individual who's familiar to the subject, and then it's gone, and then 2 pictures come up to the screen, and then they have to choose who is the same individual. Arrangement of these choices actually vertically organized, which means sometimes high rank individual located in a high place, but sometimes low rank individual located in a high place. And we don't ask actually the rank of the individual, but whenever they are looking for high rank or low rank individual, which position fit to their representation. If it's matched to their feeling, then that should be easier. But if it's not fitting, then it would be a little bit more difficult than the fitting situation.
So do you see a delay coming in then? When you put the order the wrong way round, the lower ranking individual is higher up the screen. Do they take longer to choose that one as the one they saw previously? Whereas if you get someone, an individual from their group who they recognise as being top of the tree, and you put them at the top, they get it straight away.
Right. That's exactly the case.
Why do you think they do that? What's the reason for this?
What I think is it's more efficient for the living creature to understand the information in analogy of the space, for example. So using some similar features to connect the multidimensional information, this is reduce the cost for them to process. For example, when we think about number line, just place number from 1 to 9 in your space. Then most of the people do small number go to the left, big number go to the right. And this is also kind of analogy between the order and the space.
So do you think this is then unique to animals that are social, animals that need to be able to gauge where they are in the group in terms of their seniority, their likelihood of being attacked or their likelihood of being able to steal someone else's food? If I look at other animals like dogs and wolves that are pack animals, would I see a similar behaviour? And similarly, if I were to examine animals that usually they've evolved to live on their own, I wouldn't see this behaviour.
This is a really good point. And the reason why they have this kind of a connection between two different information is probably from more basic requirement for them to reduce the cost for the processing. This could be more universal, not maybe only in the primate lineage, but also other species as well. But which type of the information can be actually treated like this? Because for the social animals, social rank is very much important for them, right? So that could be reason why they treat this rank in analogy to the space. But for other species who may not care about this rank, in that case, maybe not the case.
So we're not so special after all. Akuma Adachi there from Kyoto University. That's it for this month. If you'd like to follow up on any of the papers we've discussed, they're all freely available on eLife's website. My name is Chris Smith from The Naked Scientists, and I will be back next month with another look at what's hot in eLife. Until then, though, goodbye.
The eLife podcast from eLife, the open access journal for outstanding research in the life and biomedical sciences. Online at elife.elifesciences.org.